Hi, good day. My name is Cherilyn M. Autos, and for today's video, I'm going to be discussing all about the central dogma of molecular biology. So, what is the central dogma of biology? The central dogma of biology, or the central dogma of molecular biology, describe the three-step process which is replication, transcription, and translation by which the information in gene flows into proteins. So it explains the flow of genetic information from DNA to RNA to protein. So the DNA encodes RNA and RNA encodes protein. The central dogma of molecular biology describes the flow of genetic information in cells from DNA to messenger RNA, or also known as mRNA, to protein. So who proposed the central dogma of molecular biology? So the one who proposed the central dogma of biology is Mr. Francis Crick. So he proposed the double helix structure of DNA. It means that the information passes from DNA to proteins via RNA, but proteins cannot pass the information back to DNA. So the next one is there are three processes of central dogma. So the first one is replication, the second one is transcription, and the third is translation. So this is the three main processes used by all cells to maintain their genetic information and to convert the genetic information encoded in DNA into gene products, which are either RNAs or protein depending on the gene. So the first one that we'll be tackling is all about the replication. So replication is the basis for biological inheritance. It copies a cell's DNA, and the enzyme's DNA polymerase copies a single parental double-stranded DNA molecule into two daughter double-stranded DNA molecules. So, in other words, replication is the process of copying a molecule of DNA. So, replication is an essential process because whenever a cell divides, the two daughter cell must contain the same genetic information or DNA as the parent cell. So the replication process relies on the fact that each strand of DNA can serve as a template for duplication. So DNA replication initiates at specific points called origin, where the DNA double helix is unwound. So a short segment of DNA or RNA called a primer is then synthesized and acts as a starting point for new DNA synthesis. So an enzyme called DNA polymerase next begins replicating the DNA by matching bases to the original strand. Once synthesis is complete, the RNA primers are replaced with DNA, and any gaps between newly synthesized DNA segments are sealed together with enzymes. So, DNA replication is a crucial process. Therefore, to ensure that mistakes or mutation are not introduced, the cell proofreads the newly synthesized DNA. Once the DNA in a cell is replicated, the cell can divide into two cells, each of which has an identical copy of the original DNA. So the second one is the transcription. Transcription is the process of creating a complementary RNA copy of a sequence of DNA. So transcription is the process of making RNA of a gene. And this copy is called messenger RNA, or also known as mRNA. So transcription is a process of turning DNA into RNA to protein. So we know that you all have heard that about the central dogma, which is DNA to RNA to protein. But transcription refers to that the first part of going from DNA to RNA. Both RNA and DNA are nucleic acids, which they use base pair of nucleotides as a complementary language that enzymes can convert back and forth from DNA to RNA. So during transcription, a DNA sequence read a DNA sequence is read by RNA polymerase, which produce a complementary antiparallel RNA strand, like you have seen in the picture below. So transcription results into a RNA uracil, or U, in all instances where the DNA thymine would have occurred. So transcription is the first step in the gene expression. So unlike DNA replication, transcription results into a RNA uracil in all instances where the DNA thymine would have occurred. So the stretch of the DNA transcribed into an RNA molecule is called transcript. Some transcripts are used as a structural or regulatory RNAs, and other encode one or more proteins. So if the transcribed genes encodes a protein, the result of transcription is messenger RNA, or mRNA, which will then be used to create that protein in the process of translation. So the third one is translation. The translation is a process by which mRNA is decoded and translated to produce a polypeptide sequence, otherwise known as protein. So this is a process by which the genetic code contained within a messenger RNA or mRNA molecule, and it is decoded to produce a specific sequence of amino acids in the polypeptide chain. So transcription has a method of synthesizing proteins, and it is directed by the mRNA and accomplished with the help of a ribosome and a large complex of ribosomal RNAs, or rRNAs, in protein. So in translation, a cell decodes the mRNA genetic message and assemble the brand new polypeptide chain. So the next one, transfer RNA or tRNA, translate the sequence of codons on the mRNA strand. And the main function of tRNA is to transfer a free amino acid from the cytoplasm to a ribosome, where it is attached to growing polypeptide chain. And tRNAs continue to add amino acids to the growing end of the polypeptide chain until they reach a stop codon on the mRNA, and the ribosomes then releases the complete protein into the cell. And that is my report is all about. And thank you for listening. Have a great day.